So the next unit we are about to start talking about is cellular transport, and we've been leading up to this scientific concept by teaching you about how the cell membrane works and how molecules can move across the cell membrane in the phospholipid bilayer through the idea of selective permeability, meaning that cells can control what comes in and out of their cell. And also by teaching you about how the internal functions of the cells work to make proteins and to store molecules and get rid of waste, and how the nucleus works. And so now I think you're ready to understand this idea of cellular transport, which is the movement of molecules across the cell membrane. In order for cells to survive, they need to exist in a liquid environment. And as you can see in this picture that I've put in the background, the middle dark part is the nucleus, and the dark outer ring of the cell is the plasma membrane. And the plasma membrane needs to be selectively permeable, meaning it only allows certain molecules to move in and out of the cell. So we're going to look at two different types of transport of molecules across the cell membrane. One would be passive transport and one would be active transport. Passive transport is when the molecules can just diffuse across the membrane without using any energy. And an example of that would be just diffusion of molecules right through the lipid bilayer. You can see that in the graphic I put on the slide. And then there's also the idea of facilitated diffusion, where the molecules can't just diffuse on their own. They need that open protein channel in order to get through the membrane. Active transport, as you can imagine, active requires energy to be active. That's when molecules can move across the membrane with the use of energy. This is common when molecules move against the concentration gradient, meaning that if there's more molecules on the inside than on the outside, as this shows, and those molecules on the outside want to get to the inside, that's the opposite of what happens in diffusion. Those molecules can't do it on their own. That protein channel actually has to use its energy to open up, let those molecules through, and then uses energy to shut the protein channel. So with the concept of passive transport, meaning that the cell does not need to use any energy in order for the molecules to move across the membrane, one that we always like to start with is the idea of diffusion. And you saw this in the first lesson that you did for this unit, where we put food coloring into the water, and you saw how those molecules of the food coloring spread through the water. And that's the concept of molecules moving from an area of high concentration to low concentration. So as you can see in this photo at the very beginning, their molecules are close together, they're highly concentrated, and as time passes, those molecules spread out more. And that concept of the molecules moving from high concentration to a lower concentration is the idea of the molecules moving down the concentration gradient. That does not require any energy. They go from where there's a lot of molecules to where there's less molecules. And that is different from active transport, where the molecules have to move against the concentration gradient or up the concentration gradient, where they go from a lower concentration to a higher concentration. Taking the concept of diffusion one step further, we start to talk about osmosis, which is the movement of water molecules specifically across the cell, the semi-permeable membrane. And because cells can do this, it enables them to regulate their internal environment, so control what's going on inside the cell. And the cells being able to control what's going on inside of them is the concept of homeostasis. And homeostasis is important. It's how we regulate our body temperatures. It's how we regulate the amounts of water in our body. It's how we continue to be a consistent functioning organism. Whether the molecules are moving in or out of a cell during osmosis depends upon the type of solution that the cell is in. If the cell, and this comes back to the idea of the concentration gradient. So osmosis is a passive form of transport. No energy is required, so that means that the molecules have to move from an area of high concentration to an area of low concentration. So looking at these pictures here, a hypotonic solution is when there are more water molecules on the inside of the cell membrane than on the outside of the cell membrane. So if molecules are going to move from where they're highly concentrated to a lower concentration, the tendency would be for the molecules to move out of the cell in a hypotonic solution. In a hypertonic solution, there are more water molecules on the outside of the cell membrane than there are on the inside of the cell membrane. So as you can probably guess, in order for the water molecules to move down the concentration gradient, they would go from the area of higher concentration outside the cell to the area of lower concentration inside the cell. And when you have an isotonic solution, that means that you have an equal amount of water molecules outside of the cell as you do inside of the cell. And so the water molecules are not going to move across the cell membrane at all at that point because there is a nice equilibrium of molecules on both sides of the membrane. And finally, the last type of cellular transport that we're going to talk about is endocytosis and exocytosis. So endocytosis is when large particles outside the cell are engulfed or brought inside the cell. You can see that on the left-hand side, there's a molecule on the outside that's making its way in. And then exocytosis is when large particles are released from the cell by secreting them or expelling them out. That's on the right-hand side. That particle starts on the inside of the cell and works its way out. It kind of fuses into the cell membrane where it's then expelled out and pushed out. Endocytosis and exocytosis would be the last type of transport we're going to discuss. So just a quick review, we had passive transport, which was osmosis, diffusion, and facilitated diffusion. And then we had active transport, which would be the movement of molecules against the concentration gradient. And then endocytosis and exocytosis.